My name is Kelsey Sensin. I'm an educator at the DNA Learning Center in Brooklyn. So maybe some of you have gotten a chance to visit, or maybe some will come visit someday. I'm really excited to be here with Dr. Jeremy Borniger, who's going to talk to us about science. But real quick, I just wanted to share real quick a few things on our website you might be familiar with. First of all, our Meet a Scientist series, if you go to the DNA Learning Center, you can find that through here. But if you want to watch any old past events, you can scroll down and we have all of those recorded. So if you want to learn about some of these topics, or those are also available on our YouTube channel. This will also show us any of the talks that are coming up if you're interested in attending more of these. And then the other thing I want to highlight is that our science camp registration just opened this week. We're really excited. We're going to have science camps at our Dolan Learning Center out on Long Island. We will have some virtual camps. We'll have lots of camps here in Brooklyn as well. And then we also have a camp each week at our center in Regeneron up in Sleepy Hollow and Tarrytown area up in Westchester. So if you're interested in summer camps, check us out. And then, and also I wanted to remind people that we try to make it as easy as possible if you have financial need or from an underrepresented background to get scholarships. So as you go through the application process, there will be a place that you can indicate that if you have need. And yeah, those are my main housekeeping things. So I'm going to turn it over to Dr. Jeremy Borniger to talk to you a little bit about how cancer and disease in the brain all go together.
Great, thanks so much. Let me just share my slides. Yeah, thanks so much for inviting me to come in and talk about things that I am really excited about, which is how the brain sort of surveils the body during health and disease. And also, I'm happy to talk about my sort of journey from around high school age up towards how I became a scientist and sort of my trajectory into that area. So today my goal is to sort of dispel the notion that the brain is a separate entity from the body and replace that with the idea where the body and the brain sort of represent two partners who are in a constant sort of back and forth conversation. My lab is broadly interested in how the brain detects changes in the body and how these signals are interpreted within the brain, and then reciprocally how the brain then sends out signals to the body to effectuate change. So to investigate these phenomena, we use cancer as a tool to perturb the host system and then assess how the brain responds. So I'm going to sort of go through some background first, and then we'll go into the types of experiments that we do. But first of all, who am I? I was born in Washington, D.C. This is me as a preemie. I was 2.5 months premature. Weighed only like 3.5 pounds when I was born. And I was the second baby at George Washington University to get surfactant, which pretty much allowed me to breathe and probably allowed me to live at that time. So after growing up a bit, I went to Indiana University in Bloomington, Indiana, and then on to graduate school at Ohio State University, that's shown here. And then I went and did a postdoctoral fellowship at Stanford with Luis de la Sea, who is right in the middle here. I don't know if you can see my mouse, but right here. And he actually discovered those cells that were shown in the title slide, these ones. So it was sort of a good match. And then in 2019, I was offered a position here at Cold Spring Harbor, and I started in early 2020, right before the pandemic shut everything down. So this was the lab back in late 2020. So I wanted to also highlight that the path into science, even though you see it quite often as a straight line where someone goes to high school, college, grad school, postdoc, and then it's just a straight line. I feel like almost no one actually has that in reality, even though they like to say they do. So I just wanted to highlight sort of how I jumped around from high school on. So when I graduated high school in 2007, I was really unsure about what I wanted to do. I had no idea really what even the options were. All I knew was that I liked science more than other things. And so I could see myself sort of working in the natural world in some way, but that was pretty much all that I knew. And I was initially actually, when I started my time in college, I was interested in going into underwater archaeology. And so that was basically because I had my scuba diving license, and I had gone to a couple of underwater archaeological digs in Mexico. And this is one of them down here. This is a Mayan sacrifice that's about 35 feet underwater. So I was very excited to start doing that. However, I made it about 6 months into that program and realized that the people who were running the program and the science that they would sort of get you exposed to just didn't really resonate with me. So I sort of freaked out at that time, thinking, oh no, I don't know what to do now. But I pivoted to something sort of related to archaeology, which was anthropology. So I actually got my degree in biological anthropology, and I spent some time after college working in Uganda at a chimpanzee research site. Basically, my job there for a couple months was to walk through the forest all day, every day, and track chimpanzees and pick up their poop so that we can do genetic analyses and look at kinship relationships, also categorize their behavior. And unfortunately, a lot of my time was spent dismantling poacher traps and stopping a lot of the poaching in the region. And this was really cool. But after this, I was still fascinated with animal behavior, but I really didn't, I didn't feel like I could really ask the types of questions that I wanted to ask. So in anthropology, you can't really do any manipulation. It's all observation. And so I thought I need to go a little bit deeper and be able to manipulate the structures that control behavior. And of course, the organ that controls your behavior is your brain. And so I managed to convince a hiring committee at Ohio State that I was, even though I had not gotten a degree in neuroscience, I could then get a PhD in neuroscience. And so this is me during graduate school in the lab. And then I continued on that trajectory. After working in neuroscience for a couple of years, I then went and did a postdoc, continued working in neuroscience, but also through my interest, started working in cancer as well. And then in 2020, I started the lab here. So it was not a straight line. I just want to highlight that again. It was a rather sort of circuitous path where I jumped around from different interests to different interests. And eventually things sort of just managed to fall into place as my interests got narrower and narrower. Okay, so a little bit of background. How does the brain actively sense change in the body? So you're probably all aware of the recent Nobel Prizes that were awarded last year for the discovery of the molecular mechanisms of touch and temperature. This is from groundbreaking work from the labs of David Julius and Artem Patapushin, who made just really incredible inroads into understanding really the molecular mechanisms of how we sense temperature and touch. And these are primarily driven by the receptors that they discovered called TRP or TRP receptors. Here, TRPV1, you might be familiar with it as it is the capsaicin receptor that makes chili peppers feel hot. And this other one called piezo receptors, which actually sense stretching, not at the level of your entire body, but they sense stretching at a cellular level. So how is tissue deforming? How do blood vessels stretch? That sort of thing. And that is really important for development of your body, but also controlling the response in health and disease. And so one of the really important things is that these receptors and many others are actually located on nerves in your body. And they're on things called sensory nerves. And these sensory nerves are distributed pretty much everywhere throughout your body. They reach essentially everywhere that blood vessels reach. So I remember the saying from undergraduate physiology class, basically no cell in your body is more than 10 or 15 microns away from a blood vessel. And that's pretty much true for sensory nerves as well. So it's a very sort of thick net that is stretched across your entire body and within your body as well. And so these sensory nerves are directly connected to the brain through the spinal cord, and they sense things like pain, temperature, touch, but other things like acid and the levels of oxygen and carbon dioxide, glucose, lactate, other metabolites, inflammation, stretch, hormones, hypoxia, many other things. And it's not only that they sense things on your skin, they also sense things inside your organs. So sensory nerves don't do everything though. There's another mode by which the brain senses what's going on in the body, and that's through receptors that are present within the brain. And they sense changes in molecules that are circulating in the blood, but also the cerebrospinal fluid, or CSF. And the importance of these non-sensory nerve pathways is sort of highlighted in this picture, which is a famous picture from the late 90s, early 2000s, where you see this really fat mouse next to a skinny one. Well, it turns out that this mouse is deficient in an important gene, one that codes for a hormone released primarily by fat cells called leptin. So this mouse lacks the gene that encodes for leptin. But leptin doesn't act in fat primarily. It actually acts in the brain. And so leptin usually is a signal to your brain that you are full. You have enough nutrients to stop eating and to stop storing energy as fat. And when this signaling goes wrong, things can go way out of whack, as you see here. Leptin receptors in the brain powerfully regulate body weight and appetite in response to leptin, which is secreted by mostly fat cells. And so when the mouse doesn't have any leptin, the brain interprets it as if the mouse is starving. And so it puts the body into sort of a fat storage mode and promotes food intake. Now, interestingly, this is also observed in humans. These are a family of patients, and the patients labeled A, B, and C, you can see in the first panel, are rather overweight and obese. However, when they were given low dose leptin, so in the normal range that you and I experience, they lost almost 85% of their body weight over the course of just 1 year. So this is sort of highlighting the really powerful way that the brain senses changes in the body, including hormones such as leptin, in order to really control how the body responds to changes and how you control where the energy in your body gets stored and where it gets spent. So that's the first step, is sensing changes. So when the brain senses these changes, how does it, know what to do with all of this information that's coming up through the spinal cord and all this information that's coming in through these through the blood and the CSF? And so at the sort of most fundamental level, the way that the brain does this is through a mechanism called integration. And this is different than say like reflexes. Reflexes, which is something you might think of as a quick way that the brain responds to changes in the body, but actually a lot of reflexes don't even require that the signal make it to the brain in order for the response to be enacted. Something like withdrawing your hand from a hot stove really fast. That signal actually doesn't even have to make it to your brain before you elicit the withdrawal behavior. But things that are more complicated, such as pressure in your abdomen or something like that, that needs to be interpreted and integrated into your brain to in order for it to respond. And the way that it does this is through integration. And at the most basic level, every neuron in your brain receives input from at least one and probably up to several thousand other cells. And not all of the signals from every single input are important. So the neuron or the brain cell must decide what is the most important and what is enough for it to respond to. So at the basic level, the way that this works is that because brain cells are electrically active, they communicate with other cells via electric charge. So that's shown here by changes in voltage, which is just a difference between 2 points in electric charge. And normal neurons are around -70 millivolts. So that just means that inside the cell is -70 millivolts in relation to the outside. So there's a gradient. And so when they receive inputs from other neurons, there is a change in this membrane potential. That's what it's called. And if the change is not very strong, nothing happens. Even if the change is from dozens of other cells, if they're not strong enough, it won't elicit a change. But if 2 inputs come in, say these 2, or the same input comes in many times in close succession, they'll reach a threshold. And usually it depends on which type of neuron you're talking about, but the threshold is somewhere around -15 or -55. There's a pretty big range, but once it reaches a threshold, there's no going back. All of a sudden, this signal is now important, and the neuron will fire a big burst of electricity called an action potential. And this is essentially how all the cells in your brain interpret signals that they receive. Now, this is a sort of major area of research in neuroscience, and understanding how this process at the single cell level relates to what's going on at the level of the whole brain is a major question in neuroscience. Okay, so now we have an idea of how the signals get to the brain, how the brain integrates these signals at a basic level. But how about what does the brain do to respond to these signals? So in essence, there are 2 major pathways that regulate your body's functions subconsciously. And they both act to really restore what's called homeostasis. So all of you, I hope, at least right now, are sort of sitting here in a comfortable temperature-controlled room, and your body temperature is stable at 98.6, and your heart rate is stable, and your blood pressure is stable. And all of these things are sort of stable in a nice range that makes you feel healthy and alert. And that is really the notion of homeostasis or stability. And what the brain constantly is doing is monitoring what's going on in your body and sending out commands to different organs saying, let's turn this dial up, let's turn this dial down to see if we can return to homeostasis. And it does this through 2 different pathways. So the first one is part of what's called the peripheral nervous system. It's the autonomic nervous system, also called the automatic nervous system. And this controls everything that you don't think about, basically, such as blood pressure, how much you sweat, how big your pupil diameter gets, how fast you digest food, all these other things. And it's divided into 2 general parts, the sort of fight or flight system, which is called the sympathetic division, or the rest and digest system, which is the parasympathetic division. And these sort of operate in a yin and yang fashion to sort of to stabilize your body's functions. And there's another pathway that doesn't rely directly on the nervous system, but it's sort of part of the nervous system. And these are things such as the HPA axis, which is part of your neuroendocrine system, where it starts in the brain, but then it leaves the brain through the bloodstream. So one important methods that your brain regulates your body is through axes such as this. Here we have the hypothalamus, which contains cells that produce this hormone called CRH, which then signals into the pituitary gland, which then signals with another molecule out into your bloodstream, which then acts in your adrenal gland, which secretes cortisol and other hormones. And if you've ever heard sort of a pop science lecture, they usually refer to cortisol as a stress hormone, but it does so much more than that. It powerfully regulates the circadian rhythms in almost all of your cells in the body. It controls your immune system. It regulates how much glucose is in your bloodstream, and many other things. And so these are the two main components I wanted to make you aware of. Okay, so with all of that sort of out of the way, you should have sort of a good understanding of how the brain monitors and then reciprocally controls the body in response to a changing environment. So where does my lab come into the picture? So we think of cancer as a new challenge to the body, where the brain and the body need to detect and respond to a growing tumor in real time in order to prevent disease and ultimately death. And up until recently, the role of the nervous system in these processes has been largely sort of overlooked. And this is partially because cancer biologists and neuroscientists and clinicians, they rarely get together. They don't talk very much until relatively recently. And there was also a finding about 50 years ago now that suggested that cancer cells are electrically silent. And so that really sort of set the stage for people to ignore the nervous system for a long time in cancer processes, because everyone thought if cancer cells aren't electrically active, then they can't really interact with the nervous system. But that's not the case at all. Okay, so the primary questions in the lab are focused on understanding basically how do cancer cells interact with the nervous system? How does the brain detect cancer in the body? And then can we manipulate these pathways to treat disease or improve outcomes? And so the types of projects that we do fall into 4 general areas that are sort of increasing in their complexity. And so the first area focuses on really the smallest and finest level that we can focus in on, which is electrochemical interactions, so direct contacts between nerves and cancer cells. Then we also have what's sort of called the paracrine interactions, which just means in the local area. So how does a neuron or a nerve interact with another cell within a tumor? And then how does that dictate what happens to the cancer cell? And then how does that reciprocally mess up the nervous system? And then if you keep on zooming out, there are also these systemic neural cancer interactions. So how does the nervous system regulate cancer? And then how does cancer influence the central nervous system? And then the final area that we're starting to look into is basically all of these things combined, but instead of being focused directly on cancer, it's focused on cancer treatment, because a lot of the problems that patients experience can be ascribed to the effects of toxic treatments such as chemotherapy or radiotherapy. Okay, so what types of experiments do we do to examine the interactions between the nervous system and cancer? So I mentioned earlier the sympathetic nervous system as a main system that responds to changes in the body. And so we have some projects that are looking at the sympathetic nervous system at virtually every level of its organization. So there is a brain structure that's a very tiny brain structure. Here it's shown by these blue arrows. It's this blue spot here called the locus coeruleus. And this structure is responsible for the vast majority of norepinephrine in your brain. And norepinephrine is also called noradrenaline. It's frequently thought of as sort of a stress hormone, something that wakes you up very quickly. You ever see people, you know, in comic books getting shots of adrenaline or something to wake up? This is essentially what that is. But what's really interesting about this structure is that it not only sends projections to the brain, it also influences the body. And so my lab is very much interested in how changes in activity of this structure dictate downstream responses to cancer in the body. And so one of the ways that we do this is using this relatively new technique. It's not really new anymore, but it's a way that we can control cells non-invasively with light. It's called optogenetics. It sounds very complicated, but it's simply using optics or light to manipulate a genetically encoded protein. So this is a rat, but we do this in mice, and a fiber optic is put in their brain, and it goes down to an area like this, where we have the fiber that makes a little indent right here, right on top of this structure. And this is the locus coeruleus in a mouse. This is a human over here, and this is a mouse here. And here, what I've done is I've labeled the norepinephrine producing cells in red. So these are the cells we're targeting. And the green color is the optogenetic protein. So you can see that they overlap quite well. And what this means is that these cells are now sensitive to blue light. And how does that work? Well, when you hit these cells with blue light, the optogenetic protein that we put in them actually opens up, and it allows a bunch of positive ions to rush in. And as I mentioned before, the nervous system is an electrically active system that operates by moving different ions in and out of cells to change the voltage of its membrane. And so if you can do that with light, you can effectively control the nervous system with light. And why might this be important for cancer? Well, there was a study published last year showing one potential really interesting way that norepinephrine and the sympathetic nervous system can control something that's really important for cancer, and that's the immune system. So here I'm going to show you a video that was taken in a living mouse lymph node, where there's a bunch of T cells, which are cells of your immune system, and you'll see them moving around. But then when norepinephrine or noradrenaline, which is the same thing, is added, you'll see what happens to these really important immune cells in the body. Whoops. So here are both those immune cells moving around, and then let's add norepinephrine. Okay, and you see they essentially freeze. So do you think that these immune cells that are frozen by the sympathetic nervous system would be good at eliminating a tumor in the body if they can't move correctly? So that's something that we're very much interested to see. How can we connect this central node in the brain with processes in the body like this? So that's one of the types of experiments that we do. To give you an idea of just how powerful this little group of cells, remember, it is just these tiny little dots right here in the brain. To give you an idea of how strong these cells are in turning on this sort of fight or flight response, I'll show you a classic example of sympathetic nervous system activation, and that is dilation of the pupils. So in humans, when we're scared or frightened by something, like a sudden movement or some skittering or something that we didn't expect, our pupils rapidly dilate due to the actions of the sympathetic nervous system. And this is also true for mice, which I'll show you here. So here this is a mouse that's in a nose cone. So it's breathing in some anesthesia. So it's actually asleep here, but its eyes open. So this is the mouse's eye. The mouse is asleep, and we have a fiber optic going into its brain that you can't see. And that is controlling those cells in the locus coeruleus. that release norepinephrine. And so I'm going to hit play, and you'll see that this mouse's pupil is relatively small. But then when the laser comes on and activates the locus coeruleus right about here, now you'll see the pupil growing pretty rapidly. And this indicates that we successfully turned on these cells in the brain. And to give you a better idea of what it looks like, here's a before and after. So before we turn on the laser in this small brain structure, we see no pupil dilation, but then immediately afterwards, we see a large pupil dilation. And this is an easy to measure readout of the sympathetic nervous system. So we're very much interested in how the neurons that control responses like this can be manipulated in the context of cancer. So that's one area, but we also want to go a little bit deeper. What's going on in the local environment of the tumor and neurons? So to look at these types of interactions, it's important to sort of reduce the complexity of the system as much as possible. And we do that usually by taking cells out of an animal or getting commercial cells and culturing them in a dish. So that's what you're seeing here. These are neural cells. This is a video. Here, the purple that you're seeing is actually mitochondria labeled in the neurons. So here's a bunch of healthy neurons that are communicating with each other and moving around over the course of about 15 hours. And so what we do is we grow the neurons like this, and then we plop a bunch of cancer cells on top of them. So this is a different type of image that instead of using light to take the image, we use a beam of electrons to take the image, because at scales this small, the wavelengths of light that you need to take a picture are too big to really get good resolution. So we have to go even smaller than a wavelength of light to get these types of pictures. So what you're seeing here is a blob, or a spheroid, that's the technical term, of breast cancer cells that are growing on top of other neurons and neural related cells like glial cells. And so we're really interested in how are these connections being made between these cells, and can we manipulate those? And so to give you an idea of what this looks like in a video, here, instead of being purple, the neurons are labeled in green, and this color will sort of fade away as the recording goes on. This is about a 15-hour recording. But this little blob up here, this is a ball of cancer cells, and you'll see how they interact with the neurons in the culture. So the cells just sort of latch on to the neurons and follow them along the axons. So over the course of about 15 hours, you can see how this little blob of cancer cells really just explodes into a migrating mass of tumor cells. And we are very much interested in what are the factors that the neurons are providing that make it so these cancer cells like growing in this direction, and reciprocally, what are the signals that the cancer cells make that make it so that the neurons like to interact with them? Because I think if we can get at those fundamental questions, we can sort of keep on zooming out and see how this influences the entire host system. Okay, so here I just wanted to show some other pictures, zooming in a bit further. So this is now at 1,000 times zoomed in. These are some neural processes, so axons, right here, connecting with a breast cancer cell down here. And so we're really interested in understanding what are the molecular mechanisms that mediate these interactions, and can we manipulate those interactions to stop cancer growth. So here's a couple of examples of neurons and axons that are these long processes here interacting with breast cancer cells, which are here in these sort of elongated blobs. Okay, so finally, what's the point? What's the benefit of all these approaches? So there's currently no drugs that are approved to target the nervous system for cancer. But I think this will change in the near future. And we need a much better understanding of just the basic biology that goes into this first. And to give you an example of what I'm talking about, here's some data from a study that was published a couple of years ago in a model of prostate cancer. But it's also been repeated in breast cancer now, where the receptor for norepinephrine, which I talked about before, was deleted in the mouse. And when you give mice that don't have that receptor tumor cells, they don't grow hardly at all. So if you are looking at this panel A, this is just growth of the cancer over time. And you see that in mice that have the receptor in black, the tumor grows quite big. And you can see that over here. And in mice that don't have the receptor, it almost doesn't grow at all. And that's shown here. And furthermore, the tumors that grow in the mice with the norepinephrine receptor are very highly vascularized. These are all blood vessels in there. And the ones that don't grow very much don't have many blood vessels in them. And this is all of this effect is driven by this single receptor for one neurotransmitter. So there's a lot of room for improvement on understanding what's going on. And just to show that this is important, actually, so based on this type of research, a drug that blocks this receptor was developed, and it was shown to have efficacy in breast cancer. So we have a lot of room to improve, but there's some really promising strategies out there to really understand how the nervous system interacts with the body, and then reciprocally how that gets thrown out of whack in cancer. So with that, I'll end with some acknowledgements. Here's a snapshot of my lab from this past summer. Obviously, I couldn't have shown you any of these cool pictures without all of their work, and then the funding sources that have been generous enough to support the work. So with that, I'll be happy to take questions if there are any. And if you have questions that you don't think about until later, feel free to email me. I'm always happy to talk.